Let's now review a key feature of the cell annotation platform, which is the ability for scientists to log into the site and provide reviews or feedback of the cell annotations that their colleagues have provided on the site using the molecular data. So I've opened the main page of the data set here. You can see on the upper right that I'm logged in. And to get context of this data set, I'll look at the summary. In this case, it's very detailed. You can see that this is associated with a study of Crohn's disease with pediatric patients. It looks like there were 14 patients initially sequenced, then there were 8 matched biopsies, and 13 biopsies were used as controls. These were non-inflamed functional gastrointestinal disorders. Looks like over 200,000 cells were sequenced, and maybe about 4,000 less passed through QC. If we look at the cell types here, as expected, you'll see many immune cells within this tissue. Some of the cell types I can see are related to just endothelial tissue, but there's also cell types here which are distinct to the ileum, the small intestine. So let's look at the molecular data. So I've opened the molecular data page. I can see the embedding, the heat map of gene expression and differentially expressed genes. And we can go through the workflows of what users might do for providing feedback. The first thing that comes to mind is that we have these T cells here. It's the largest amount of cells within this data set. And we might look how the scientists have defined these T cells. They've used T as just a way to quickly annotate these, and they've provided more context. T cell as the closest ontology term in a biomedical database, synonyms, and they've defined T cells very, very broadly. So they've included things that I would consider to be very strong synonyms closely related to a T cell, like T lymphocytes, with a hyphen and with not. They've also included subclasses of T cells, like immature T cell, mature T cells. They have marker genes here. If need be, I could see context for these marker genes. Let's see, this is CD3E. Canonical marker genes, and then some sort of rationale of why T cells were annotated in such a way. So let's actually just select these. And you'll see this on the embedding. It's this large group of red cells here. And if need be, I can see already these were the marker genes that were used to define these T cells. I can get a closer look at the marker genes here by opening the heat map. And I can, let's say, TRAC is here, and then CD3D, MALAT11. This is great. So some feedback I might give, and this is fairly common, is that these are actually a fairly broad group of cells. It could be split up. And so in this case, I would disagree. So I will select the cells again. And given I'm logged in, I can provide feedback. This is not my data set. It's data set of my colleagues. And I can do this with any data set that's published on CAP. You can see I have agree, disagree, or I don't know. In this case, I want to disagree. And I would just say that this is a group of cells which should be split up into more precise cell types. So let's say this is split. And you'll see that there are prompts given that provide the user with some context of how they could disagree. In this case, I think there are, let's say at least 2. There are 2 cell types that should be provided in this case. And there is some hint that maybe I could split this for mature T cells and immature T cells. And so, as opposed to giving a very broad cell type of T cells, and I can see for some reason T cell is here as well, we should tell the scientists that actually you should really split this up into canonical mature T cells or immature T cells. And those are 2 groups. So the first group we would have, I've done this before, so we'll see that this is immature T cell, or we could use mature T cell, and common markers that are used here are things like CD4, which will come up, there we go, and maybe something like CD8A, there we go. And there we go, so that is one group, mature T cell, another group would be, let's say, immature T cell, and a common cell marker here would be CD3D, which you can see up here. Okay, an explanation is something like, this is too broad of a group of cells. This cluster really should be split into the 2 main categories, subclasses of T cells. immature T cells, immature T cells. My lab uses the marker CD4 for this, and there is clear evidence in the heat map above for D, which is a marker for immature T cell. And then I can send my feedback. And this will show me the feedback. This will be the feedback that will be shown to other scientists, and that is saved. If need be, I can look at the other feedback, which we'll get into. And you can see my feedback was recorded right here, which also we'll cover when we go through other, when we just summarize how to view all this feedback. So that is an example of a disagreement. An agreement, let's take a look at a cell type which is related to this. Well, I actually don't know the cell type outside of the small intestine. So those are Paneth cells. We can see how the scientists define Paneth cells. Like me, I don't know the synonym for a Paneth cell, and these are the markers that were used for Paneth cells. So in this case, in this example, I'd like to provide feedback, and I'd like to agree with this. So for Paneth cells, I can see that they're right here, but let's select to see where they are. And I will select the Paneth cells. Now they're selected, and I can provide feedback. And in this case, I would like to agree. I would like to say, my lab also defines Paneth cells using the marker. And I'll send my feedback. This will then show my feedback. There we go. And that was an example of agreement. We could also do the same thing with I don't know. In some case, it could be, let's say, natural killer cells, which we study a great deal. I can select these. These are how natural killer cells are defined. If we select the natural killer cells, see this is where they are in the embedding. Provide feedback is I don't know if these were annotated correctly. Right, and I can send my feedback for this as well. This will show to my colleagues that I don't know about this. Another example would be actually disagreeing with scientists how they define the cell types themselves. So we can go back to T cell here. In this case, it's related to this notion of a T cell is the same thing as mature T cell or an immature T cell. In this case, I would probably disagree with this, and then I might also disagree that track would be used for as a marker for T cells. It's actually a very specific type of T cell. So in this case, I would refine the metadata. So I think that immature T cell is really not a synonym for T cell, mature T cell is not a synonym for T cells, and track is a particular type of a T cell as well. And that would, I can send some feedback regarding why it is that I disagree with the definitions, and that will be saved as well. So if one wishes, one could see all of the feedback in the dataset. So we can go back to the dataset main page, and I can look at all the feedback that I have provided. So in terms of T cell, I've disagreed with the molecular definitions, and I've also provided some feedback of how the scientist has defined those T cells. I've recorded that I don't know regarding how natural killer cells were defined, and then Paneth cells, I express my agreement. There's ways that we can sort this page if there was lots of feedback that was provided here, as opposed to the example. We can also see, you can imagine if there's other scientists providing this feedback, sort of globally where there's agreement and disagreement, and this is simply coloring the embedding. So to see where there's agreement so far, if you recall, I provided agreement here in the Paneth cells, I provided disagreement with the T cells, and I said I don't know about the natural killer cells. And that's one way that we can view the embedding. And as more and more votes are accumulated, you'll be able to actually see a spectrum of the agreements. But so far it was just me reviewing this data set. So that is the rough workflow of how to provide feedback, or to think of these as reviews of the cell annotation